Okay, hello everybody. Welcome to this live by Zoom tutorial on using Ingenuity Pathway Analysis IPA at the University of Manchester. So we have a license, a single concurrent user license. So that's why I have to run this as a demo, just so I can show you in real time how to use the software. The link for the usage instructions is about to come up as I show you the screen and we go through the different topics that we'll be covering today. So let me just share that screen and then we'll have a quick look at what we're going to do today. Firstly, just to use our license here in Manchester, you need to, we run with a booking system. So I'll just make sure everybody knows how to use that. Then, yeah, basically just install and run the software. I won't be spending much time on that at all. And loading your data into IPA, that's quite important. I mean, some people have come through the core facility and we usually make a session for you anyway, but if you want to load up some other data from elsewhere, then we'll go through that. Then there's running the analysis jobs in IPA. Then there's understanding what's actually been done there, understanding the statistics, the numbers that IPA uses to rank results. Interpretation of the analyses is next. So we'll just go through what IPA has to offer. Then I'll give a few tips on using the interface. And then finally, analysis match, which is the new feature. Okay, so starting at the calendar usage. So this is important so that people don't get frustrated. If you want to work at a particular time, it's important that everybody sort of adheres to that. Otherwise, you try and get on your session and you can't get there, and you've set time aside. So we all need to know how to use that. So I'm just going to share that screen that I've got, which is my calendar, just as an example. So let me share my calendar and share that. Now, as you can see, my calendar is a mess, and that's because, and in fact, I've even made it more simple because a lot of people share calendars with me, and so it's just all, we just have a whole lot of different things in there. The calendar for me at the moment, that's Ingenuity usage, is the blue ones. Okay, so if you were to make a booking, say I wanted to make a booking here on Friday, then when I click on the calendar, it's going to automatically want to put it in my personal calendar, which if you put it in there, no one else is going to see it. So we want to scroll down to Ingenuity usage, and then you put your name in there. And what we're suggesting now is you just put your full name, not any contact details, just for for privacy, etc. So that's the way to do it, and then everyone will see it as long as it's in Ingenuity usage. At the moment, it's fairly heavily used, as you'd expect. We're in a lockdown, so we are adhering to our etiquette, which is that you can book 4 hours a week. There's a few nuances to that, but read the instructions. I'll point you to those. Install the software. I'm working from Windows. I think it works quite straightforwardly from a Mac as well. Linux is what we tend to use in bioinformatics, but at least when we checked, that didn't work too well. So we tend to work in a Windows VM. So if you go to that link, you can install the software, and then you'll need your university email address plus your IPA password, obviously not your university password. And that will get you into that. Okay, so if we now look at IPA, so I'm going to share that screen again. Share. Okay, so hopefully you can all see now that my IPA screen. As you can see, I have lots of different projects in here. So when you want to start a new project, it's a good idea to make a new folder for that. So in this case, I'm going to make a training one. So I tend to use the right click, but with all these software, you can get to what you want with different ways, but I just do right click, new project, and then we'll call that training 2020. Okay, so when you look in there, you'll see it automatically, the software bar automatically will populate it with these different folders here. And these are to do with the different types of analyses that you could be running on your data. So the first thing we're going to be doing is looking at how you load data into. So this is about loading data. So firstly, what kind of data? Now, obviously, I tend to mostly analyze RNA-seq data, and so that's what I'll be talking about. But you can load anything. You can load a list of proteins. You can even do metabolites, but it tends to be genes and proteins, and you can have any kind of data with that. Sometimes you need to try and trick the interface a bit, but generally it's expecting things like a fold change, a log fold change, a p-value, and a false discovery rate correction. So anything like that, but obviously now what I'm going to do is just what most people, most of our users at the moment have is RNA-seq data, what that looks like. So let's consider a situation like this where we have 3 groups. So we have a control, we have someone who's marginally ill, and we have someone who's very, very ill, and these are A and T. So if we had an RNA-seq data set, perhaps we would have something like this. So in our first comparison, which is T versus C, so T versus the control, we'd have a log fold change, a p-value, and a p-adjusted value. And then we would also have, say, A versus control, and log fold change, p-value, p-adjusted value, and then we'd have a long list of genes there. Okay. So the first hurdle that you're going to face when you load up your data is, what does IPA understand by an observation? Now that can be slightly misleading, because for us as biologists, you might think, well, control is an observation, treatment A is or situation A is an observation, and T is an observation, but that's not the way this works. An observation is one of these comparisons in IPA. Okay, so that's the first thing. That's kind of, it's a jargon to do with this software that's slightly counterintuitive, in my opinion. Okay, so an observation is where we generally have compared 2 things. Now, if you had a proteomics experiment and perhaps there was a lot of preparation on the prep, and so that you know specifically it's the secreted proteins under this condition, and you've just got that one condition, then observation makes sense. You would just load up that. So, and it would just be sort of presence or not of a certain protein. So in that case, observation is kind of, it makes sense to call it an observation. But often what we're loading up is actually a comparison between 2 things, and IPA calls that an observation. Okay. So I'll run through loading up the data. IPA. Okay, so in this case, what I'm going to do is upload a file that looks like the one that you just saw. It has names on the left and two comparisons. So we'll go to Data Set Files, right-click, Upload Dataset. Okay, now I need to navigate to where my file is. Okay, here's my file. Okay, so this is what the file looks like. So I had names, plus remember I had these two comparisons, log for change, p-value, p-adjusted, etc. So we just have to now assign the data. So we'll just call that an ID, and it already knows that it was a gene symbol, but you might have to pick that. It's processing here, so it's trying to identify these names and link it to some kind of instance in its own database. Okay. Hello, what happened there? Okay. So now we're going to go through this process, and now why it's important to know what an observation is. So remember that these three columns all belong to the same observation in the terminology of IPA. All right, my internet is gone. Okay, so then we have to check. So IPA tries to guess the identity of these things, but it's made a mistake here. It said that was a p-value. It's going to have to change this to be a p-adjusted value, or false discovery rate, and we'll do the same here. Okay, we can also change these names here. Instead of having observation 1, you can make it something that makes more sense. So if you go to edit observation names, our observation 1 is T versus C, and our observation, so this one, oh, I don't know what's happened to my internet. Okay, so we'll call these T versus C. T versus A. Okay. All right, so that's all we need now. As you see, after trying to identify, it's actually identified 32,000 of these genes here, and I identified them with its database. So we can now say save. Just going to have a look at the chat. Somebody's asked a question. Do you need to have adjusted p-values? No, you don't. Can you run your analyses with just fold change? Yeah, you can have just fold change, or you can have fold change and p-values, etc. Okay, so when you've loaded it up, then this is the screen you get. And what you want to do is go here to analyze the data set. Okay, and the core analysis, that's the one, basic one we're going to talk about today. It's the one where most of the functionality of IPA rests. Okay, so we'll put that on. And at this point, it asks you what kind of data set it is, expression analysis, and this question here is to do kind of with the Z scores and the coloring. We'll talk about that later. The Z scores and the coloring of the images in terms of up or down regulation gets chosen with this. So fold change. So if you don't have fold change, only p-value, you won't get that. Everything will just be the same color. But if you've got fold change, up and down will be different. Okay, so so you just say next on that. The defaults were correct. And so at this next step, what we're going to do is decide how many genes are in our gene list. In other words, which genes are significantly changed. Now, data sets differ. I know the biology, trying to get those numbers right in terms of how long do you wait? Do you wait till there's a late response and maybe thousands of genes have changed? Or do you take an early response where there's maybe only 50 genes that are statistically different between conditions? In a way, for this IPA analysis, we shouldn't get too hung up on the numbers. So we don't really care too much about p-value or fold change. I happen to have a data set here that fits nicely where I can, so I've loaded up log fold change base 2. So when I put 1, that's a 2-fold change. So I'm saying anything that's smaller than a 2-fold change or greater than a 2-fold up. And I'm going to put p-value here. Again, this is a topic. If I now hit recalculate, it tells me how many genes passed filter on these two comparisons of ours. Okay. Now, what we want is around 1,000, but IPA says anywhere kind of between 200 and 3,000 is okay, but 1,000 is definitely kind of a sweet spot for this. You don't want too few genes so that IPA doesn't have enough data to kind of do the analysis, but you also don't want a significant chunk of the whole genome. So that's why you don't, and in fact, IPA won't accept over 4,000, I think it is. So 1,000 is about right. But as you see, we got both of them are being assessed with the same setting here. That's the way this works. And that gives me quite a nice number. I've got 1,500 and 800, so that's fine. And then we'll say run analysis. And then I always give it some kind of logical name. So p-value 0.05, log fold change of 1. You can add more notes here if you want, just to remind yourself what you actually did. But that, for me, I always find that's fine. Okay, so that's, we're just going to have that running away now in the background. And what I would like to do is now tell you about how the statistics of enrichment. Okay, we need to know this, and that's running in the background. Okay, so understanding the statistics of enrichment. Hang on, I just need a little drink. Okay, so now statistics, for some people this can be quite scary, but I'll try and make it as I understand it, which is quite simple. I'm not a statistician. I just try to learn enough to get by. So let's imagine what we've got here is our long list. Remember, there were 30,000 odd proteins. Okay, this is our long list. And with our particular cutoffs, we've got our hot list up here. Okay, and let's say, just for argument's sake now, while we're trying to understand what enrichment means, say that's 1,000 genes. Okay, so what we want to know is a pathway like this, is it enriched for my genes in here, right? So we want to know what pathways are quite highly represented with this list here. Okay, so the way this works is as follows. We take a list like this and we'll just flatten it into another list. Okay, so we've got a long list here, we've got our sub list, and then we've got a list here. Let's say this is 10. I haven't exactly counted them, but let's say it's 10. Okay, so when we talk about enrichment, so I'm going to go into this in detail because it's really important that people understand this. Okay, so this is really important. So we'll go really slow, and if there's any questions, please do ask, because this is important that you understand this thing. And all of IPA kind of rests on this particular point, this enrichment step, okay? Everything revolves around this. It's important that you understand it. Okay, so when we say enrichment, what that means is enriched above a random list of genes. Now, we don't have to be perfectly random, because we've actually got we've uploaded all the data. So we've got all the possible genes that we could have measured in our data. If you got proteomics, that could be, you know, just all the secreted proteins, something like that. And then you've got your sublist. So we don't have to look at all the proteins then. But in this case, we've got all the known genes in that. So the way you can imagine this analysis works is that since our list here is 1,000, IPA can run multiple times 1,000 genes that we pick at random from this list. So not based on any biology, just simply pick 1,000 genes. How many times is there a hit in this cardiomyocyte differentiation pathway? Okay. And what you'd expect is something like that, a distribution like this. When you run this over and over and over, you will find, okay, it can't be lower than 0. So there'll be times when there's just no hits. There'll be times when, you know, maybe the most frequent number of hits you get is perhaps 3. I'm just guessing now. And every now and then, very rare case, you might get 10. You might find all of them when you, in your list of 1,000, might contain all 10 of these genes, and that might just be random. So when we run this over and over, we can get some kind of a distribution of how many times we get a hit in this list from a list of 1,000. Okay. But then you come with a real life, based on good sound biology experiment. We've got a list of 1,000, and when we run this, we've only got one chance, of course, of running this, and it turns out maybe we get 9 or 8 hits. And so when you've got a distribution of expectation, you could say, well, if there was 9, that's, I tend to not get that very often. And so IPA can then put a p-value to that. Okay, so that is done with this particular test, which is the right-tailed Fisher's exact test. What came up here, what I meant to say is that IPA has lots of pathways. So this list here of 10 would be this list here, but this could be a pathway of apoptosis, this could be cell cycle, etc., etc., etc. You've got a whole long bank of pathways and lists of proteins to compare to. The second analysis that happens at this point is what they call a Z-score, which is to do with the directionality. So if you did add a fold change, a log fold change, IPA has a well controlled data set where it could say if most of these genes are upregulated, that suggests that this particular pathway is in an activated state. Okay, so that has to do with the Z-score. Alternatively, it could be an inhibitor. Sometimes genes are upregulated, it means something's inhibited. But the point is, it's linked, the fold change can be linked to the biology. And sometimes things don't agree. The log fold changes don't agree with each other in terms of this pathway, or this pathway has been well studied in one tissue, but you're studying a different tissue. So there's, you know, there's things that don't always match up. All of this needs to be taken with a pinch of salt, as I'm sure you're going to hear me say again. It's just to help you come up with hypotheses. Okay, so that's the important thing. When we talk about enrichment, we talk about enrichment above or beyond or in comparison to a random list. Okay, so there's a kind of a false positive kind of screen right in there, and that's why I've got no problem picking p-value. I don't really care if people take FDR, but I think p-value is okay, because we have got in the second step, when we're studying enrichment, we've got a kind of a control for false positives because we're comparing to a random list, which is by definition a false positive. Okay, so that's how to understand the statistics of enrichment. Now we'll have a look at the interpretation of the analyses. These are just what IPA will present. Oh, hello, I managed to draw on this. Okay. That's not my daughter, by the way, that's me. Oh, it's disappeared again. Right, let's get IPA back. IPA is still running, but we won't worry about that because I've done this before. And here we've got another one. Okay, analyses. So when IPA has run, we'll end up with, okay. Right, so here we've got an example analysis that we're going to look at. So here we ran it with, remember, p-value, log-fold change of 1. And we can look at either one of these. So let's just open one of those. Okay, so the settings that you used are to be found here. So p-value and log-fold change, those were the settings. And this is how many genes came through. So if you forgot what you did or you didn't write it down, you can always find it there. Then you get a sort of a some highlights here. But all of these top canonical pathways are under the tabs here. So you can drill into all of these, but you could get a quick overview here. If something looks interesting, you can go straight to it. Otherwise, you'd work your way through these. So in this point 6, I talk about canonical pathways to networks. So with canonical pathways, that's kind of what I showed you. You'd have particular pathways. Which ones has IPA what has IPA assessed as being the most enriched? And then we get you get ranked by p-value. It's the minus log of, because when something's significant, it would be a small p-value. So if we put a minus log in front of it, it becomes a big number. So that's here. So the height of these bars is to do with the p-value. Now remember, I mentioned z-score to do with, it relates the fold change to the activation state of that particular pathway. And the coloring here tells you what we're dealing with. So a positive z-score is related to an activation. A blue is related to an inhibition. If it was white, don't see any white ones here. Basically, the fold changes are not agreeing with each other. And it comes to a z-score of 0. If it's gray, then simply IPA does not have information available to be able to say one way or the other. Okay, so if we click on the bar, we get a list of proteins that were in your list and that are also in this pathway. And then if we go to open pathway, we get those nice plots that IPA is famous for. Okay, so that's what that looks like. Now we've got a different color scheme. So remember, orange is for a positive z-score activation. Blue is for a negative, so inhibition. When it comes to these diagrams, then we're looking at the fold change of the individual genes. Green means a down regulation. Red means an up regulation. So in your data that was uploaded, that's what these colors are referring to. And a brighter, the more intense the color, the higher that fold change was. When you see a double line around something, that is a complex, and you can expand the complex. So if you right click, you first have to highlight it, right click, you can show the members. I'm not going to do that now. I don't want to spend too much time on going through this interface now, because it is my 0.7. So we'll come back and talk about how we can work with this, these interfaces more. I just want to go through the whole, the list of tabs here. Now, upstream analysis. What I'm going to show you is my last. Now remember, when we did this analysis here, then we had a list. All this was here. Turns out it's just a flat list here, right? A list of genes, a list of proteins. And we're saying, is it enriched or not? And what IPA does is say, it's got some data sets, public data sets, and it's saying, when I perturb an upstream regulator, that upstream regulator could also be a drug. Say I add a drug to a cell, published data sets say, 50 genes are changed. Those 50 genes can just be another list like this. So when we run the analysis, we run the core analysis. If we see a good enrichment or an overlap between this list that came from a downstream activator, upstream activator, it's downstream of upstream activator, IPA can just say, yeah, that's enriched. And then we can report that as not the list, but the upstream regulator. Okay, so it's exactly the same principle. It's just where that list came from is a different place. It's not a pathway. It came from an experiment where we perturbed an upstream regulator. So in this case, we've got IFNB1, and there's a good overlap here. That would be the p-value that we're talking about. The z-score tells you whether the upstream regulator so your fold changes whether they're consistent with the upstream regulator being in an activated state, or in this case, this regulator is in an inhibited state. So when you inhibit this one, does an inhibition, then the gene list that comes out looks like the gene list that it's just received from you. Okay, so that's what upstream regulator is, kind of taking one step back. Not just the pathway, but taking one step back. Now, diseases and functions. This actually looks a lot more like canonical pathways. Again, we've got particular diseases, and this is quite hierarchical. So, of course, under cancer, we've got different kinds of cancer, and each of those cancers will have a number of genes that are known to be players in that disease. So if disease is your area to work in, this one is worth looking at. And again, everything that applies the same way. Okay, we've got regulator effects here. I don't know too much about that, so I'm just going to leave that alone for now. What I want to do is give you this networks. So networks, it works on a different principle. So if you imagine I'm going to stop the shared screen just because I'm probably, to do this properly, I should have made actually a separate diagram, but hand waving might help a bit. So if you imagine IPA's got a database that's very highly curated. That's why we're paying all this money, because there's actual scientists who are being employed to read papers and find relationships between entities, between chemicals and proteins, genes, etc. When they find an important finding that they think needs to go in the database, they discuss it with a team of people. If the team of people agree, then it goes into their database. So what we're paying for is the intellectual property of all that work. Okay, it's not just text mining or something like that. This is all done by scientists who are, experts in the field, who are able to make these assessments. So if you imagine they've got this huge database that's got all these dots in it, which is all the known proteins, and then there's links. If you imagine there's a ball and stick, there's all these links, and each of those links means something. It's a relationship, And in fact, they even keep the paper that information comes from. It's all in there. So we've got this vast kind of, you could think of a protein or a gene universe with all these dots and lines joining them all up. And when you come with your list of 1,000 proteins, on the fly, an analysis happens. And what I mean by that is not taking known data from the past in terms of pathways. It's just taking all these binary, so binary by that I mean one protein, another protein with a link between it, which makes this huge network. And it tries to find within that huge universe of proteins and genes, it's trying to find a little network, a little cluster in there that's heavily populated with your proteins. Okay, so this is a mathematical, there are different mathematical solutions to this. You can be quite lenient on different aspects of this. Some of the issues are, we don't want, if there's 2 of your proteins, but there's, say, the path between those 2 means you have to go through another 2. That's quite a long distance. So you would prioritize where there's direct connection between the 2 proteins that were in your list. So there's all ways you can assess that. And so you can try and grow the network so that it includes as many of your genes as possible. But you can also prune it, limit it, so that the paths between your different proteins doesn't get too long. So when you do that, you can then end up with a pathway, with a network. And let me share the screen again. I need to get back to IPA, so hopefully you're seeing IPA again. So these are the different mathematical solutions that could have been made. So what that looks like, I'm just going to show you an example here, is something like this. And as you can see, because all of these genes, proteins are colored, that meant they were all in your data set. And this is one of the solutions that it came up with. Now, as you can see here, there's actually, IPA has also said what it thinks that network is involved with. And that's because once it's made this group of well-connected proteins, it then runs another kind of core analysis on its canonical pathways and tries to annotate what that sublist is involved with, if you see what I mean. So it's not using this information to build the network. The network is done mathematically, and then we try and annotate it. But that means for you, you can look through, find what's interesting. Okay. So that's networks. Pathways is just ways of making the network. Let me see if I can do that. I think it's under Path Designer. You know, when you hit that button, you get something that's just slightly like a painting. So let me do that so you can see it. Anyway, that's the kind of thing it looks. Oops, okay, it's not happy with that. But anyway, when you do that, you can get a slightly different view of your data, which you might prefer when it comes to doing a presentation, etc. Okay, so as you see, I'm skipping through some of these, but we have a certain amount of time to deal with, and there's so much I could go into here, and some of those things I don't know either. Then there's also this analysis match, which is our new feature, and I will be talking about that at 3. So we've still got 10 minutes now to go through working with this IPA interface, as I like to think of it, because the interface is kind of shared amongst all these. If you click through on any of these, you get to this kind of an interface. So for example, what we did was we clicked on a death receptor and we got this. So I'm just going to give you a few tips now on working with this interface, but the same applies for networks, or for any of these interfaces that where you've got these kind of ball and stick type things. Right, so what we did see was when you highlight a complex, then we can expand that. So if I say show the members, okay, so these are the proteins that make up that particular complex. Only one of them was in your list. And that's why I'm just going to highlight and delete that. You can see it, I don't know if you can see that, but it's sort of gray to red. So it's trying to indicate that some of the components of that complex are there. Now, remember what I did there, I expanded it and then I just deleted it. So we can do that with all of this. If we don't like some of this pathway, we could just delete aspects of it. So we can just select those and I'll hit the delete button and it disappears. So we can simplify this pathway. We can just drag stuff around. You can reformat it and design it as you like. Now, another valuable thing when it comes to writing your paper, writing your thesis, when you highlight the link, you can get some information. So if you double click, it tells you something about that relationship. Okay, so these are different proteins. The line is a relationship, and the relationship here says in cytoplasm, flip protein increases inhibition of caspase. Okay, and you can say, well, prove it. And if you click that hyperlink there, it opens your browser. I don't know, again, whether I'm going to actually see this or whether you're going to see it. Maybe somebody can just give me a nod if you do see it. But anyway, what I'm seeing here is a reference to a paper, Microbiological Immunology, and the year that this paper was printed. Okay, so by doing that, you can just go through and interrogate all these relationships yeah. Okay, so you would have noticed there's two other buttons here. One of them's build, one of them's overlay. So I'm going to look at overlay here. And the one thing I'm going to show you, so there's a lot of things you can do here, but what I want to show you is I want to overlay. Remember this one we're looking at here is A versus C. But what about T versus C? It's in the same experiment. I might be interested to know what T versus C looks like on this particular pathway. This pathway came up in A versus C, but what does it look like on T versus C? So I'm going to say add more, and now I can pick any of mine here, but obviously it doesn't really make that much sense. So I'm going to pick one that's from the same experimental context. Okay, and it's immediately updated it, and oh, it looks quite similar. So we can just flip between the two. So you can see the difference. So there was a lot in common. My slightly sick person and my very sick person have got, compared to control, the same kind of things are going on. So, you know, you can see that could be quite useful, especially if you've got a lot of comparisons. You might have 6 different comparisons, and you can just sort of scroll through on an interesting pathway that you found, and you can just compare that. Okay, so that's canonical pathways. What I'm going to show you on networks was a build, because there was a build here. So when it comes to build, what you can do is start to grow and add things. Okay, so let's say we're interested in this protein here, HES7, and for some reason we're really excited about that protein. What we want to do is grow. So what we want to do is know, is there any other proteins perhaps in my own data set, even though this network analysis has been run, maybe there's other proteins in my data set that are known to interact with this one. So we're going to say, let's look at direct partners. Let's get all the molecules that are, let's say, downstream of this protein here. Okay, so if we hit apply, as you can see, there were 3 proteins, 3 known, but it doesn't look like any of them were in your data set, or at least not in your significant list of 1,000. If they were, they would have come up colored. Okay, so that's the way we can grow a data set. And in fact, it's how you could even build a whole network just from one protein. So let's say we want to do that. So let's say we're just going to take this one protein. I can also search for them up here. Let's say I'm just going to get rid of all of this. Oh, hang on. I think I did something weird there. Okay. Apologies, IPA. Okay, so that was quite interesting. Let's see if I can delete that. No, I can't. I can just, oh, I think let's start again, right? So let's just close this. I think the IPA software is busy screaming at me right now. Okay, let's go back. Let's view this network. Oh, let's do something else. Let's just start. No, I'll change my mind again. Let's hit view network again. Okay, so I happened to hit enter and I meant to hit delete there. So there we are. So let's pick all of these. Okay, so let's say we're going to start from 2 proteins and we want to build a network around them. So we're going to select them and we're going to say build and grow. And then we'll say all direct. And let's just, so that we don't get the situation of going crazy, but it is a dangerous setting, we just say, give us 10 up or downstream, and we'll say apply. And there you go. So you could add a couple of proteins, and some of them are even in our list. And so you can just build your own network in that way. You can build out. So if you've got one protein of interest, you want to see who it interacts with, what it interacts with, you can slowly keep expanding. Okay, so in this way, we could go on for quite a lot longer, but let's leave it there for now. So that's essentially a few tips. That was number 7 in our point 7 tips for working on the IPA interface. Okay, so let's take a little bit of a break. Just gonna have a cup of tea. So before we get into analysis match, I don't know, were there any questions? You could switch your mic on if you want to. If you had any questions about anything that we've done there, just maybe just going through what I just covered. Quite a lot of topics there.
Would it be possible to go back to the grow that you just performed?
Okie doke. Here, right, yep.
Do you mind sharing the screen, please?
Oh, sorry. Share the screen. Okay, yep, there we are.
Yeah, so where you go on limit molecules to use Ingenuity knowledge base, would it be possible to select use molecules from a separate data set or analysis that I performed previously?
Yeah, Yeah, it says also list. So yeah, you could, I've never done that, but so then it comes to lists. So then working with lists is, if we go with this analysis that I added here, so there's these lists here, right? So at any time, you can select proteins, select these and say save as list. So if we come here, if I select those, there should be a save as list under here somewhere.
I think it was the second one, the second icon.
Was it that one? Yeah, add to my list. Okay, so then you would have a list there. I've tried working with lists and sometimes it's a bit frustrating. So just have a go with that, but at least you'll know.
It's more data sets. I was talking about.
Sorry, say again.
I'm more interested in talking about the data sets, not lists. So
Okay.
using molecules from a data set or analysis to sort of combine networks.
Yeah. Well, I suppose it looks like it's possible, but since I've never done it, and given our time at the moment, I don't want to sort of pursue this too far. But it looks like it can all be done, and and it's also important to know where to look. So you could always, if you go down that route and you get caught, you could either email me or email your IPA support people. And they could show you how to do that or point you to a tutorial on how to do that. Yeah.
Okay, great. Thank you.
Okay.
Can I ask a quick question? In your summary, if you press summary, before canonical pathways.
Oh yes, yep. Sorry, yep.
What do p-values? What does overlap percent mean? How significant? What is the 5.0%? Is it significant?
Well, that's for p-value, you mean?
No, I mean overlap, percent overlap.
Okay.
There is different percentage.
Yeah, I think. Well, so p-value is statistically significant, but the overlap will be, I guess, how many proteins within that pathway overlap with your, well, overlap with your list. So in this case, it's 12%, and so that's dependent on. So the significance will depend on how many components there are within a particular pathway. Because if we had a pathway of, say ribosomal proteins, okay? So maybe there was a ribosomal protein and there's 800 there. The chances of finding a ribosomal protein in any random list of 1,000 is going to be much higher than a pathway that has maybe 10 components. Okay, so the chances of finding one gene that belongs to, I don't know, say this tweak signaling has 10 proteins in it. If we pick 1,000 genes at random from our long list, the chances of one being of finding tweak signaling in that list will be less than a ribosomal protein, because that is much more. So that's why the percentage overlap doesn't strictly correlate with significance of p-value. Yeah, it has to do with that distribution that I showed you. If we were dealing with ribosomal proteins, then when we pick a list of 1,000, it's probably going to end up being somewhere like 50. Yeah, that we on average we tend to hit 50 ribosomal proteins in a random list. Yeah, whereas on a smaller set, that number is smaller, and that affects the significance.
Thank you.
Yeah, I hope that makes sense.
And no bacteria.
Sorry?
May I ask another question?
Okay,
or
let's
do you
take
need to
out
move
the
on?
last one for now, and then we'll get on to analysis match. Yep.
Yes. So when you're adding your list with A versus, what was it, A versus C and T versus C, you have log fold change, and then you have your p-value input. Aside from the overlay function where you are allowed to kind of toggle back and forth between within a specific network or pathway, and see how specific components of that pathway are changing between your two observation groups. Is there another functionality within IPA that allows you to assess, let's say within observation 1, which genes are significantly upregulated or downregulated that are also not in observation 2?
Yeah, okay, so that's quite a good lead into the next section. So
okay, perfect. Thank you.
that's quite good. So let's move on then to analysis match. So we're now on the last point here. And right, so let's go to IPA. So the basis of analysis match, there's a few bases for this. I need to explain to you what makes analysis match different to other things. The first point is a comparison analysis. So that relates to the question that was just asked. Okay, so here we've got a comparison analysis is one of the options. You see, we had the data files where we loaded up, we ran an analysis, and that got all plonked into there. There's something here called a comparison analysis. So if we go to a new comparison analysis, so we need to understand a comparison analysis in order to understand analysis match. So that's why I'm going to explain this, apart from the fact that there was a question about it. Okay, so at this point, we need to pick what things we want to compare. Yeah. So we're going to have this analysis that we've done. And so we'll say, we'll just add those 2 points like that. And then we could just hit view comparison. Let me just get rid of that screen. Okay. So this is a little bit small, but what we have here is, and there's different ways of ranking these different groups here, right? So, and there's different ways of filtering it. So you could say, well, I only want things with a significant p-value that are different, etc. So there's various settings that you can change for that. But essentially what it tries to do is tell you, with this kind of heat map approach, which pathways are showing similar behavior between our 2 observations. Remember, going right back. So we've got our observation T versus C and T versus A in this case. I think that's meant to be A versus C and T versus C. I think I must have labeled these wrong. Okay, so it was T versus C and A versus C. Anyway, whatever, it doesn't really matter about the labeling at this point. But the point, you can see what it's done is the things that have a similar behavior. So in other words, these had a positive Z score, orange. That means they were significant and they were activated. Whereas here we have significant and inhibited. So these colors are matching. And there's different ways, like I said, of ranking this. Here it looks like it's ranked it by Z score. You can do a sort of a hierarchical clustering where all the things that are, you put the inhibited, so Z minus Z score together, and all the things with a plus Z score together in that way. So that's just different ways of organizing this comparison. Okay, and then also within there, all that what we've discussed before, so upstream upstream analysis, networks, etc., is also done as part of this comparison. Yeah. So that's quite powerful. I haven't used it that much, I must say. I think it's the kind of thing that is kind of, if you're a IPA user and you've got a real data set, you want to, and you've got lots of conditions that you had compared, you'd really want to drill into that. You might find something quite interesting. I'm just going to close that for now. If I was to save it, it would land up there. I didn't, when I did my core analysis, I set that analysis going, because I showed you that, I didn't tell it specifically to run analysis match. So analysis match just emerges up here, okay? And what analysis match does is it takes public data sets, and the reason I was searching down here is I thought they were all available, and I think they are, it's just I'm not in the kind of heat of the moment, I'm not finding them, but it takes public data sets, and again, I'm going to stop the share and do my hand waving because I don't have a separate slide for this. Okay, so we've got public data that's available to IPA, and there might be some cases it's not public, but they bought the data perhaps from, you know, the Monsantos, etc., drug companies, that sort of thing. They bought the data in, and what they do is they take an analysis. They might rerun the analysis comparing, say, conditions to produce these observations, and they take the top 1,000 genes by significance. Okay, top 1,000. So they're not going on particular p-values. Remember what I said? It's actually the you want that sweet spot of the numbers of proteins. So it's taken the top 1,000 in any one of their comparisons in these public data sets. They've run a core analysis, and then they've done a comparison analysis. Exactly what we just did when we compared those two observations from our own data. They're comparing our data with analyses from public data sets. So remember, with comparison analysis, what we were comparing in this comparison was the canonical pathways or the networks or the upstream regulators. So it's not a gene by gene comparison, it's the IPA results that are being compared. Yeah. So there's a few concepts there. So we take 1,000 genes from any comparison in our public data, we run an IPA analysis, and then we compare the results of that analysis to our own analysis run on IPA. Okay, so hopefully that makes sense. I should really have a slide for that. That will be my next job for the next time I run this demo. So let's share the screen again. Yeah, right, share. So what's interesting is the top hits were the cases in the past, and it's kind of what you'd expect because it's exactly the same result, except that IPA gets updated every now and then. But when I've run these demos through the years, those end up as the top hits, which is not a surprise, right? So we should be getting the same. We should be getting the same result. So when we run a comparison analysis between our A versus C, then it's going to hit. It's going to find the closest match. Yeah, and that's what we get in this score here. But of course, what's interesting is public data. Yeah, I mean, you know, you might find something here. You might find a public data set that you just completely didn't expect. Yeah, so we've got a multiple sclerosis data set here. Now, I'm going to try this again because I'm absolutely sure I can find the data in here somewhere. Ah, it's under libraries. That's why. Okay. So here we go. It's under here, under libraries, and then we've got this omics soft and various. So if we look here, this one's called, say this multiple sclerosis, peripheral blood. So that's probably not there. Anyway, it's one of these lists under here, and you can find it back. But if we click on that, let's just see what we're going to get. Okay, so this is with the analysis run with that particular data set. So you could just interrogate that data set now. You could go through it just like it with your own data, find out which canonical pathways there were, etc. And you could run a comparison analysis, but I suspect it is there somewhere. I'm just not finding it. Oh, evaluate, view as heat map. There we go. So if I highlight the whole thing, then these things light up. Let's see, view as heat map. That's probably going to look like our comparison analysis. And there we go. Okay, so, and we're still stuck on our hierarchical clustering. Let's go back to score. But, and it seems to have found upstream regulators. That's what it's reporting. But the point is, the comparison analysis that I showed you, which was where we'd compared our two experimental conditions, now are done with this other data set. That's a public data set. Okay, so I think in a way, for now, that's all I've got to say. So just to wrap up, here's our list of topics that we've covered. So using our license, obviously you need to install the software locally, how to load your data, how to run a job, understanding the nature of this analysis, and then the various outputs, and then some tips on using the interfaces, because the interfaces share a sort of similar principles. And then we had a brief look at analysis match. Okay, so thanks very much for listening in, and good luck.